All right, 2700, a snow day at the university, but that's not going to interrupt our discussion about all of the invertebrates that we've got to spend time talking about. Today, we're going to talk about a bunch of disparate, and we're uncertain, enigmatic and disparate spiralians. Spiralians, of course, are this group within the protostomes, the sister group to the ecdysozoans, and they contain the annelids and the mollusks. And today we're going to talk about some Gnathiferans, so some jaw-bearing groups, including the chaetognath worms and the rotifers. And then we'll finish off beginning to talk about some of the Lophophore taxa, the taxa that contain this streaming Lophophore. So first of all, the rotifers, little bags of jelly with circular wheel organs at the interior end. They're triploblasts, they're bilaterally symmetrical. They're in Brusca in chapter 13. And the ciliary corona on the head, so you're seeing a whole bunch of them over here. This is typically, the taxonomists call this a wheel organ. It's the word rotifer is from the Latin for wheel. So this wheel organ sets up currents, and those currents bring in the little tiny particulates that the organisms feed on. Some of the other apomorphies for the group include this unpaired retrocerebral gland that secretes mucus that lubricates the wheel. And then the female has this vitellarium, this syncytial structure down here that produces eggs. They have a syncytial epidermis, so a multinucleated, sharing kind of an epidermis. And their reproduction, even though they're gonochoric, they can reproduce parthenogenically. We'll talk a little bit about that. So first of all, the outline of the musculature and how the organism, these little bags kind of move around. They can swim. The ciliary organ can actually move them around and they can swim, but they can also kind of inch forward. And they do this because they have these alternating, like we've seen with so many other groups so far, circular and longitudinal muscles at right angles, but then they also have transverse rings, and it's squeezing upon this closed hemocoel within the body. So they have a layer of this circular muscle and longitudinal muscle. So just like we've seen with some of the jellies, that across a closed liquid repositions the body. So you can imagine squeezing the balloon that you can squeeze, and this little inching bit of plankton will move around. The body wall can actually be quite hard. So there's what's called an intracellular lamina. And a lamina, so it's produced, this keratin-like protein that's produced as an endodermal skeleton. And depending on how hard that endodermal skeleton is, it can actually become quite hard and become what's called a lorica or a lorica. And that can be quite a hardened thing. So you're looking at two different kind of branched up, almost like wings or teeth of a lorica on this particular species of rotifer there on the right-hand side of the slide. So they're not hermaphrodites like we've talked about with a lot of the groups so far. They're gonochoric, they're males and females. They can reproduce parthenogenetically. When the males are around, just like we saw with some of those flatworms, there's a hypodermic impregnation. So there's no kind of sharing or specific part of the body of the female. So there's just the injection of sperm into the coelom. And there is an alternation in many of the species that we know about. There's an alternation of sexual and asexual generations. Males are quite aberrant. Rupert Fox and Barnes, who like to wax lyrical about all things invertebrate, said males, if present at all, are dwarf, aberrant, short-lived, and little more than a swimming testes and an oversized penis. Now, one of the other amazing attributes of the rotifers is that they can undergo cryptobiosis. So for upwards of a century, although I put Tribbiani quotes around this, and it hasn't actually been tested, it's been described, that they can be dormant and then brought back to life with the application of water. They're quite amazing at understanding or withstanding large changes in temperature, and upwards of 200 degrees Celsius is the claim, and large doses of ionizing radiation. Now, recently, a couple of years ago, these two researchers showed that different bedolid rotifers are highly resistant to this ionizing kinds of radiation. They did this in PNAS. And so they're capable of withstanding doses that result in for all other species, results in hundreds of breaks and breaking up of the DNA. And these researchers hypothesized that this resilience to ionizing radiation was a characteristic of their an adaptation of their evolutionary capacity to survive repeated episodes of desiccation that they would encounter in their freshwater environments. So locomotion and feeding, the contraction, we talked about this, the alternating layers of muscle. So contracting the circular musculature stretches the body. So there you can see behind my floating head, circular musculature. And the longitudinal muscles running top to bottom here. So squeezing it stretches out the body, and squeezing the longitudinal muscles stretches out. So it stretches, it retracts, it stretches, it retracts. And the pseudocele, so this is acting as a hydrostatic skeleton that permits the action of these right angle muscles to reshape this soft-bodied organism and allow it to move through space. So as I described, the ciliary organ, which you can see moving through in wheels here, sets up these feeding currents. But one of the other ways that the rotifer is, the characteristic movement of the rotifer is this telescoping of the body. And that's accomplished, that's the alternating muscles, the longitudinal and circular muscles that allow it to kind of inchworm through space or through their water environment. As you saw in some of the earlier pictures, the wheel organ can assume a bunch of different shapes and sizes, and that's associated with different roles, different ecological roles that they take up in their environment. Some of them are quite simple filter feeders. Some of them are raptorivores and move around and capture things, are predators, predatory, and some of them are omnivores. So moving on to the rotifers, to this other enigmatic taxa, the arrowworms or the chaetognaths. So in your book, in the Brusca et al. Text, this is called the enigmatic protostome. So arrowworms aren't impressively diverse, only about 150 species, marine. They have the apomorphies for this phylum include these grasping spines up at the top of the head that are made of alpha keton that are hollow. They capture prey and envenomate that prey. They have ciliary fences, which we think are all about the sensing prey. These are mobile predators. They have these lateral fins that you can see running up and down the body, and a stratified epidermis, so that there's a chitinous cuticle up at the top end of the body, and then all along the rest there's 3 to 5 layers of epidermis. So the grasping spines, you can see of 2 different individuals there. These ciliary fences just behind to sense the prey. And then the lateral fins. So the root of the word chaetognath, chaeta is Greek for long hair, gnathos, remember, is jaw, so hairy jaws. Found in all marine environments, they're major predators and major sources of major players in marine food systems. But they're quite small. They have no discrete gas or excretory systems. And we know because of these ciliary fences, we have quite a good fossil record from just during the Cambrian explosion. So in the famous Burgess Shale of Western Canada, they're full of the chaetognath worms are a major predator within that fossil bed. And so long phylogenetic history, only about 150, we think, extant species. But unfortunately, our understanding of them, our contemporary understanding of them, has been contaminated, for lack of a better term, because lots of the intra-phylum phylogenetics early on that happened were the result of contaminants, just like we saw with the xenocoelomorphs, where a small marine, our understanding of the phylogenetic position of this small marine predator was reduced because early phylogenetic systematists analyzed prey item DNA rather than the DNA of the predator itself, in this case, the chaetognath worm. This picture here, this is a fossil. So this is a contemporary worm on the left-hand side and a fossil on the right. You can see this, it's a strategy. The chaetognaths arrived upon the shape of the grasping jaws at the front, hairy jaws at the front with ciliary fences, and it's been successful for hundreds of millions of years. So this is the floating through here. This is a paper from last year that looked at chaetognaths in a food web just off of Mexico. And they have some great descriptions of the roles that they play. So the chaetognaths here in the center, fed upon by a large number of upper level in the trophic sense fishes, feeding upon a vast variety of crustaceans and other parts of the plankton, and then themselves, this lower part of the food web indicating that they themselves are food for a large, diverse assemblage of parasites. So they're chaetognaths themselves being voracious carnivores that prey on a diverse type of zooplankton, including mollusks, crustaceans, fish, and fish larvae. And then they themselves are consumed by zooplanktophagous predators like cephalopods and juvenile and adult fishes. And they have, because of this, they're quite important players in a marine food web, in a marine environment. So a couple more pictures of these apomorphies for the group, these large grasping spines at the front end. And then look here at the small gift. So the chaetognath there, this video is going to sit and move through. So sit and wait predator. So a little copepod kind of swimming by, don't move, don't move, don't move. Now what I was mentioning about one of the recurring themes that's come up in this course as we've talked about the phylogenetic position, our understanding of changes in the phylogenetic position of major groups or taxa within groups, is the role that phylogenetic loss plays. That there'll be characteristics, apomorphies or synapomorphies of higher level groups at the phylum level, that members within that will, they'll be characterized by the loss of that characteristic. So this has happened quite frequently here. So this is a phylogeny of the known families of chaetognath worms. And on the right-hand side of the slide, what you'll see circled here in orange are across this matrix of different traits that characterize the group. Everything in orange is a loss. So it's a group where the successful strategy that they arrived upon just in the Cambrian explosion is then whittled away through time, and so there hasn't been so many additions of different strategies, but more so the loss of some of the original ones. Now, as you practice drawing this simplified version of the Dunn phylogeny, what you've come across is that we have the protostomes, the ecdysozoans and the spiralians, so arthropods, mollusks, and annelids here in the spiralians, rotifers, this is where we're talking about right now. But this polytomy here, this chaetognaths, the position of chaetognaths has been uncertain. Are they an earlier branching lineage of protostome? Are they a misplaced spiralian? This is one of the ongoing kind of enigmatic mysteries of protostome or of invertebrate phylogenetics. So Dunn et al. Talked about this in their paper. If you've gone through CourseLink and had a look at that Dunn paper, you know that they kind of fall onto the side of interpreting chaetognaths as a deep branching lineage of protostomes. And they do that because it's consistent with the early appearance of these grasping spines in the fossil record, and as well as the presence of some deuterostome-like features, including deuterostomy, intracily, and radial cleavage. Developmentally, that makes them much more like a deuterostome and less like the remainder of the protostomes, the spiralians and the ecdysozoans. But enigmatic, so here's a look. So this is your Dunn phylogeny spread across. We've got different traits here from the basement membrane, from different nerves and a brain cephalization, and then across to a through gut and the mesoderm. So you can this is maybe a good picture, a good part of the PDF to pick up from when you see these lecture slides later on, to map out across the phylogeny where the appearance, or loss, secondary loss of these different traits has occurred. And so chaetognaths placed out here with a lot of the characteristics of our early branching arthropods. So a basement membrane, yes, they have an interior-outerior nerves. There's lots of cephalization. But then through gut mesoderm, a coelom, a blood vascular system. These are more characteristic of some of the other groups, but their placement of the deuterostomes rather, but their placement in relation to some of the other protostomes has been ambiguous. Until about 2 years ago, when these two F-based researchers sequenced different Hox genes. So Hox genes are these, are conserved transcriptional regulators that control early and fundamental processes of embryonic development. And so they compared Hox genes across a lot of multiple phylum, and they inferred phylogenetic relationships from differential similarity of these within these Hox genes. And what they found was interesting in how it provided a link to these gnathiferan or big complex jaw-bearing protostomes. Rotifers, so these non-segmented but triple-blast, bilaterally symmetrical wheel organ having that we talked about, have a very characteristic Hox gene that until this paper was published was thought to be unique to the chaetognaths. And so the collective appearance or the sharing of this Hox genes suggests that chaetognaths, rotifers, and some other smaller phyla that we'll talk about soon are gnathiferans, and so chaetognaths then would be nested. So there's the figure from their paper. So there's the presence of this singular Hox gene. So if you kind of look at the matrix, there's none of the other ecdysozoans or the other spiralians, and certainly none of the deuterostomes that had these set of Hox genes other than the gnathiferans like the rotifers and the chaetognaths. So this is a major change to one element, one kind of polytomy within the Dunn phylogeny, simplified Dunn phylogeny, that I've asked you to figure out how to draw. So here, ecdysozoans, spiralians, uncertain placement of the chaetognaths, then the deuterostomes, and then xenocoelomorphs is the earliest branching bilaterian, and then the asymmetrical, radially symmetrical or lacking symmetry or biradially symmetrical remainder of the earliest branching animals. So this Hox gene analysis basically removes the chaetognaths and places them in from outside an uncertain placement and puts them right inside the spiralians. So chaetognaths, we can now say, you can find other former 2,700 people and say, we know about chaetognaths now. They're protostomes, they're spiralians, and they're gnathiferans. So this large Dunn phylogeny, where the chaetognaths are in here, we can now alter slightly and bring the chaetognaths inside with the rotifers. This isn't going to change how I would expect you to draw the phylogeny. You can still draw the original Dunn 2014 interpretation, but I might ask you about it in an exam to say, so what is our contemporary understanding of the chaetognaths? So the Lophophorate taxa, what are their relationships within the spiralians? So first of all, what's the Lophophore? So the Lophophore is this crown of an organism characterized by this crown of hollow ciliated tentacles that encircle the mouth. And there's an upstream collection and movement for suspension feeding. And each one of the tentacles is a hollow space that's part of the central coelomic space. So you can see on this video, with the rhythmic beating of the cilia, the movement of small particles into the interior mouth. So water is pulled down into the center of the Lophophore via this ciliary reaction and then expelled between adjacent tentacles after the front cilia have removed the food particles. So there's a cross-section here through the tentacle showing, so dice the tentacle out here with the ciliary reaction moving it down and in. And then if you look at the cross-section, there's coelom, there's body cavity. So part of the body cavity within each one of those tentacles. And a close-up video from Vimeo that's showing you some of the beautiful nature of this ciliary action. So other characteristics, so there's multiple taxa within the spiralians that have lophophores. And historically, we grouped them together and said, we talked about the lophophorate taxa. So they had this lophophore. They tended to be sessile, sedentary suspension feeders, and without the presence of a distinct head, a low degree of cephalization. And the relationship between the lophophorate bearing taxa is one of what Dunn called one of the four or five remaining few important questions in animal phylogenetics with regard to their placement within the Spiralia. So remember, a couple of times we've talked earlier about Spiralia being more or less equal to Lophotrochozoa as an older phylogenetic word. And that has to do with the placement of the Bryozoans, a Lophophorate bearing taxa that we'll talk about in a second, and how related they are to the other Lophophorate bearing taxa, the brachiopods and the pharonids. So here you can see in this left-hand hypothesis that Andreas Haenel supports that Lophotrochozoa would be roughly equivalent to Spiralia, to all of these things. This other alternative hypothesis that Nensdal et al. Groups them together and says that Bryozoans, brachiopods, and pharonids, the Lophophorata, are part of a larger group within the Spiralia called the Lophotrochozoa. So this was discussed in the Dunn paper in 2014 as one of the key remaining questions in animal phylogenetics. Last year, just this past year, there was a paper in Current Biology that came out that looked at multiple different transcriptomes from across Spiralian taxa. So you can see the outgroup here is the ecdysozoan. Then we've got a matrix of characters, morphological characters from spiral cleavage, possessing a coelom, a trochophore larva, a larva in general, a protonephridia, so what kind of, and then different Hox genes over here. And what they found was, again, a large proportion of the Spiralia, of an enigmatic or a confusing Spiralian phylogenetics is due to the frequency of loss of all of these characters, so a secondary loss or inferred loss. But what their analysis showed was strong support for what in the Dunn phylogeny was that right-hand hypothesis, linking together the pharonids, worms, the bryozoans, and the brachiopods, so all of the lophophorate-bearing taxa characterized by not having much of a head, low degree of cephalization, sedentary sessile feeders with the lophophore. So an older word that we're finding contemporary support for. So rather than splitting up, as this Andreas Haenel phylogeny on the left-hand side supported, the contemporary support seems to be here. So Lophotrochozoa is a subclade of Spiralia as we map out the lophophore. So we're going to jump chapters and talk now about the taxa, the bryozoans, the brachiopods, and the pharonids, pharonid worms. So the bryozoans, or moss animals, there's about 5,000 species of them, freshwater and marine. Moss animals makes me think of this cartoon character, but we've actually, so this is a picture you've seen before of fossils from the Silurian limestone in Guelph, and there's a bunch of this structure in these pictures that are actually fossils of bryozoans. So it's a successful, just like the chaetognaths, being a bryozoan has been a successful strategy for a long, long time. So you can see here some of the base. We talked about this figure already, but mapping the base with a basement membrane, with nerves, with a small degree of cephalization, with a through-gut and an anus and a mesoderm and a coelom and a small blood vascular system. So moss animal, but relatively complex. So the mesosome, or this is comprised of the coelom, that is contained within the lophophore. So you can see there the coelom here at the base of these kind of fruiting bodies. These are the little polyps of the bryozoan. The metastome is most of that body, and then there's a beautiful part of the organism called the zoocoelom. So this is the small exoskeleton that's secreted by the metastome, the exterior of the body. And the zoocoene, the root of this zoocoene, the zoocoene is what's preserved. That's why we have a good fossil record. And it's also lots of species characteristic traits, or lots of things here that taxonomists can measure to figure out what it is that they're looking at. Zoocoene, the root of that word is animal house, which, if you haven't seen John Belushi in his prime, makes me think of him. Animal House is the name of that film from 1978. So linking John Belushi to a particular taxon of Spiralian might be an small exam question. So like we've talked about, Lophophore taxa tend to be sessile with this U-shaped gut, and they secrete these casings or tube. And another quote here from Rupert Fox and Barnes talking about this long, tortuous taxonomic history of trying to figure out whether the Lophophore was something that was homologous or convergent across the multiple taxa, the phoronids, the brachiopods, and the bryozoans that are characterized by that. So an older Rupert Fox and Barnes diagram here, of a cross section of a bryozoan. The closest one in the Brusquedal is 17.6. But you're looking at parts of the mesocele or the metastome that, like I said earlier, go out and reach out into the tentacle. The metastome, that's the rest of the body, and then the zoocoene, that's being excreted by the metastome and creates this animal house, this casing where they live. So apomorphies for the bryozoans include this anterior part of the body that we call the introvert, which can, within which the Lophophore and the tentacles can be withdrawn. A funicular system, they can be colonial, multiciliated cells, and this colemopore here, this ciliated groove along which eggs move and are released into the environment. The funiculus is this ligament or tendon that connects the stomach to the bottom of the body wall. There's again lots of staining here on the right-hand side showing the retractor muscles that can fire across the length of the body. So there's the funiculus at the bottom here. And then the retractor muscles that can evert or pull back and retract the Lophophore depending on whether there's feeding or the escape by a predator. So they have the introvert up on top here, and then these parietal muscles that change the coelomic pressure depending on whether they're compressed or not. If they're firing, the Lophophore is averted, and if they're not, it's retracted. Now, this beautiful video came up on Twitter a little while ago, and I wanted to share it with you. So they do have a through gut. Remember, that was one of the characteristics that we mapped across the dummy phylogeny. And so a mouth in the center here, and then an anus outside of the tentacles. And what you'll see right there is the emergence of bryozoan poop shared with you. Technology is amazing. It's a wonderful time to be alive. So they live, as I said, colonially. They're physiologically integrated zooids where they share parts of the coelom. And there's polymorphism, just as we saw with some of the colonial other organisms that we've seen. There's different zooids that play different roles. So there's autozooids that feed, and there's heterozooids that are non-feeding that can be used for defense or detection of other, perhaps from locomotion, protects to help clean. So these autozooids here, feeding zooids, and then heterozooids like this avicularia, this jaw-like raptor-like thing that snaps across little fishes to move them away, and then the vibracula that might be used to clean. The colonies can take on different forms and shapes, and we've described them when different bryozoan taxonomies speak to each other. They talk about whether they're stolonate or hydroid-like or foliaceous or leaf-like or fructose or fruit-like. But just as we described sponges earlier on, these are not phylogenetic terms. They're just a description of the shape of the colony in space. So these encrusting ones are one of the most common. This is a lot of the fossils. So this is a contemporary living animal or bunch of animals, these polyps. You can see them reaching out here, and the Lophophore going out, and the ciliary action kind of drawing the food particles into the central mouth. But the zoocoeum, this is what would be fossilized. This is the kind of shape that you would expect to see of a crusting bryozoan in the Silurian limestone that your university is built on. So there's integration of the individual polyps, the individual zooids that form the colony within these different capsules. This is an experiment that was done. Similar experiments have been done on the field course, the marine field course that takes place each summer, where zooids in one part of the organism are fed a food with a Congo dye, something that dyes red. And you can see areas within zooids that are dead where there is likely no feeding taking place. But as you add, as the dye is added, it spreads throughout. It's added in one place and then is shared throughout the colony. So these might be dead little cells, or they might be cells that are non-feeding ones that have different roles, reproductive roles. Multiple kinds of larvae. There's a corona with a ciliary band for swimming and locomotion. They also have these characteristic siphonaut larvae that can live for many, many months. They're planktotrophic and not lectotrophic like these other ones. So they have a functioning gut and actively feed as they move about the water column. So they're an extremely, not only are they settling in an encrusted part like that kelp forest, the video you just saw from the micro world, but they're an active part of the plankton. There's a close-up of the siphonaut larva. They can also reproduce asexually. So there's a asexual budding from a first settling zooid, the ancestrala, that then can produce these statoblasts that are very similar to what the gemmules of the sponges produce. So little escape pods in time and space that can move away and travel long distances and be released at quite high densities. As we move through the course, particularly throughout the comparing and contrasting the two protostomes, and then comparing and contrasting protostomes and deuterostomes, the words and the phrases that we have for the different larval types may become complicated or confusing. And so I wanted to point you out that in course link, I've taken a table from one of the alternative texts that is on reserve, the Petchnik text, that has a table that goes through and details a lot of it gives you a nice kind of layout of, say, sponges, which have this kind of characteristic kind of larva called this, and a characteristic adult called this. So there's pages and pages of this that might help you array how you're thinking about the phylogenetic distribution of these larval types. I'm not going to do that today. And that may be where I stop. So thanks for being patient with this first experiment with the floating head and the blue screen as we try and take advantage of the snow day and not lose place in our trying to talk about 34 different animal phyla within only one term. So enjoy the rest of the day, and we'll see you on Friday.